Welcome to today's presentation, titled Reframing Autism. Today we will be exploring a systems biology approach to emerging biological and metabolic therapies. This presentation marks a paradigm shift in how we understand and treat autism spectrum disorder, moving away from viewing it as a strictly behavioral condition and symptom management, and instead looking at the complex, interconnected physiological systems of the whole body. Let's begin by contrasting the historical approach with modern scientific understanding. The traditional view maintains a purely neurobehavioral focus. Under this model, autism is diagnosed purely via behavioral assessments, example ADOS-2, treated primarily via applied behavior analysis, relies on symptomatic pharmacotherapy, lacks disease-modifying biological treatments. However, the systems biology view reframes autism as a multisystemic physiological condition. This modern approach recognizes underlying gastrointestinal, immune, and metabolic dysfunctions, acknowledges biological origins beginning in utero, therapies target systemic homeostasis, improves neurological outcomes via whole body health. Instead of just looking at the brain, we must look at how the gut, immune system, mitochondria, and metabolic pathways all continuously interact. If autism is multisystemic, where does it originate? The pathophysiology of ASD lies at the intersection of genetic contributors, environmental contributors, and neuroinflammation. Specifically, this intersecting crisis results in four core areas of ASD pathophysiology: synaptic defects and altered brain structure, systemic immune dysregulation, severe oxidative stress, mitochondrial and metabolic dysfunction. These are driven by candidate genes like RELN and SHANK3, prenatal environmental exposures, and chronically elevated pro-inflammatory cytokines like interleukin-1 beta and interleukin-6. We will now break down these core dysfunctions, starting with Domain 1, the gut-brain axis and the pathophysiology of intestinal permeability. This process unfolds in four distinct stages. Gut dysbiosis. We observe high levels of Clostridia and severely low levels of protective bifidobacteria. Mucus barrier degradation. The loss of mucin-degrading bacteria disrupts mucus turnover, resulting in a thinned and vulnerable intestinal barrier. Intestinal permeability. Damaged tight junctions allow undigested food peptides, such as gluten-derived gliadin and bovine beta-casomorphin, to cross the gut mucosa. Systemic immune response. The upregulation of pro-inflammatory cytokines, specifically IL-6 and IL-8, creates systemic inflammation that travels upward and crosses the blood-brain barrier. Knowing this, how do we intervene? In this, we evaluate targeted nutrition, specifically looking at two approaches. First is the GFCF diet and the opioid excess theory, which removes dietary sources of peptides that act as opioid-like molecules when incompletely digested. Clinical observation. Caregiver reports show notable reductions in stereotype behaviors and hyperactivity following strict elimination. Second is probiotic recolonization. Targeted L-plantarum and L-acidophilus supplementation significantly reduces harmful Clostridium clusters and elevated D-arabinitol levels. Clinical observation directly correlates with reduced GI distress, improved stool consistency, and improved behavioral concentration. Moving to Domain 2, we address the systemic energy crisis caused by metabolic and mitochondrial dysfunction. A healthy cellular state efficiently processes energy, maintains redox equilibrium, and produces sufficient ATP for high-demand neural networks. In stark contrast, the ASD dysfunctional state is overwhelmed by severe oxidative stress and ROS, reactive oxygen species, depleted glutathione levels, and impaired methylation. Results in a systemic energy deficit mimicking neuroregression. To address this energy deficit, we utilize comprehensive metabolic support, such as the Spectrum Needs Protocol. This is a 33-ingredient multi-domain intervention. It is broken down into three core pillars: mitochondrial cocktail, featuring L-carnitine to ferry fatty acids and coenzyme Q10 as a potent antioxidant. We must make sure that all foundation nutrients are present in sufficient amounts, then add other specialized nutrients via liposomal delivery for optimum utilization and better compliance than multiple pills and capsules. We will also utilize glutathione, NAD+, methylene blue, MOTS-C, and SS31 peptides. Comprehensive nutrition, utilizing activated folate to cross the blood-brain barrier, alongside vitamin B12 and D3. From a foundational perspective, we must ensure adequate protein, magnesium from multiple forms, B-complex, iodine, zinc, copper, vitamin C, and essential fatty acids to balance the omega-3 to 6 ratio. Neuroprotection, providing magnesium as an NMD, a glutamate antagonist, plus zinc and activated B6, and several of the other nutrients already mentioned. This is not just theoretical, it yields real-world results. Let's look at two case profiles. Case profile Big Zach, a 24-year-old with autism, cyclic vomiting, and severe pain. After an intervention of a high-dose targeted mitochondrial cocktail and chat gene targeting, the clinical outcome was profound. Pain-free, completely weaned off opiates, and vomiting ceased. Progressed to speaking in complete sentences. Case profile Carter, an 18-month-old who experienced regressive autism and lost language skills following a basic mitochondrial cocktail and TRAP1 gene targeting based on DNA sequencing. The clinical outcome was a neurological awakening. Highly talkative, socially and emotionally engaged, sensory integration issues resolved. Our third domain examines neuroinflammation and a fascinating clinical occurrence known as the fever effect. The phenomenon. Up to 80% of parents and caregivers report a dramatic temporary amelioration of stereotypic behaviors, lethargy, and inappropriate speech in children with ASD during periods of naturally occurring fever. The biological mechanism. Fever raises the internal core temperature, which directly upregulates the stress proteome. This triggers the release of heat shock proteins that regulate multiple cellular processes in the CNS. This cascade counteracts neuroinflammation, temporarily improving long-range cerebral cortical connectivity. We cannot constantly induce fevers in patients, but we can mimic this effect using saunas and phytochemicals like sulforaphane, derived from broccoli sprout extracts. Through transcriptional upregulation, sulforaphane activates the genes controlling the stress proteome. This yields three direct benefits: counteracts systemic oxidative stress and electrophile toxicity, boosts severely depressed glutathione synthesis, suppresses pro-inflammatory cytokines and neuroinflammation. Ultimately, this mechanism restores cellular homeostasis, improving synaptic connectivity and reducing aberrant behaviors. Clinical trials strongly support this. In a trial utilizing an 18-week daily oral dosing of sulforaphane versus placebo in young men, we saw remarkable efficacy. ABC score. Participants showed a 34% reduction in aberrant behavior checklist scores. SRS score. Participants showed a 17% reduction in social responsiveness scale scores. Clinical global impression. 54% of treated participants showed much or very much improvement in behavior, compared to just 9% in the placebo group. Crucially, the study revealed a withdrawal effect. The insight here is that 4 weeks after discontinuing treatment, total scores on all behavioral scales regressed toward pretreatment baselines, confirming the active, continuous biological mechanism of action. Finally, we look at advanced cellular therapies, specifically mesenchymal stem cells, or MSCs, umbilical cord-derived MSC exosomes, and their secret ohm for systemic repair. MSCs act as independent repair engines across the blood-brain barrier through immunomodulation, suppressing pro-inflammatory cytokine production like IL-6 and IL-17, neuroprotection, activating microglial repair and restoring structural synaptic integrity, the secretome paracrine effect / paracrine action, releasing neurotrophic growth factors across the BBB, metabolic support, enhancing mitochondrial biogenesis, and restoring systemic redox homeostasis. Not all stem cells are the same, however. This diagnostic matrix illustrates the current status in ASD research. MSCs, mesenchymal stem cells. Multiple early-phase trials show safety and potential efficacy. Umbilical cord-derived MSCs, Wharton's jelly. Emerging evidence of safety and high potential benefit. Autologous cord blood, investigated in clinical trials with mixed/inconsistent efficacy. HSCs, hematopoietic. Highly invasive graft versus host risk, rarely used. iPSCs, induced pluripotent, tumor risk. Used in research, not yet applied clinically. Therefore, MSCs and umbilical cord-derived MSCs currently represent the safest and most viable therapeutic paths. When we look at overlapping biological crises in ASD, gut dysbiosis, oxidative stress, neuroinflammation, and synaptic dysfunction, a clear picture emerges. As the matrix shows, targeted nutrition helps the gut, while metabolic support, phytochemicals, and cellular therapy provide varying degrees of support for the mitochondria and the brain. The core insight is this: no single intervention acts as a panacea. Each modality addresses specific nodes within the larger pathophysiological network, necessitating a combined approach. Which brings us to the future of autism care, a whole-body protocol. The next era of treatment relies on systemic homeostasis over symptom management, moving beyond behavioral containment to reversing underlying cellular crises. Multimodal synergy, combining gut-healing diets, high-dose mitochondrial cocktails, and inflammatory modulators for compounded clinical efficacy. Personalized biomarkers, utilizing DNA sequencing, microbiome mapping, and metabolic testing to match specific therapies to highly specific patient phenotypes. In conclusion, autism is a diverse biological spectrum. The next era of treatment relies on diagnosing and treating the unique cellular network of the individual. Thank you.